Welcome back. We are going to move on into the nervous system with this lecture. We're going to take a look at how nervous tissue functions. We're going to look at the different cell types, things like that, before we actually jump into the actual parts of the nervous system. So we're going to divide the nervous system into central and peripheral. So the central nervous system is the brain and spinal cord. The peripheral nervous system is everything outside of that, which consists of all of the nerves that are in your body. So central nervous system, fairly straightforward. Peripheral nervous system, we've got a little bit more going on here. Because all of these nerves are out here in your body, you're going to have information that needs to come into the central nervous system, and then there's going to be information that comes out of it. So we have multiple divisions here. So in the sensory division, we have what's called a visceral sensory. This is sensory information coming in from your organs. And then a somatic sensory. So this is your first introduction to the word somatic. When we're talking in the nervous system, somatic means essentially basic or not special in any way. So when we hear somatic, we're going to think of things like your bones, muscles, joints, basic receptors in the skin, things like that. So you get sensory information that comes in on all of these nerves, and then the peripheral nervous system talks to the central nervous system. Central nervous system is going to make some type of decision about what we want to do about the stimulus that's coming in. And that decision will then go out into the peripheral nervous system on the motor division. The motor division has a somatic division. That's the one that we've already discussed quite a bit in the muscles. So we know how the central nervous system gets a muscle to contract. So that part we've already looked at. The other side of the motor division is what's called the visceral motor division. This is also known as the autonomic nervous system because it's what's going to control all the unconscious components of your body. So we're going to be dealing with cardiac muscle, smooth muscle, and gland secretion. Within that, you've got two divisions, a sympathetic and a parasympathetic. So we are going to look at each one of these at some point, but first we're just going to talk about the tissue as a whole and kind of how it works. So as complex as the nervous system is, we can boil it down to three overlapping functions. You've got sensory information that's going to be picked up out here on your peripheral nerves that is going to move into the central nervous system. So all of the stimulus that we're picking up out in the peripheral nervous system is going to move toward the central nervous system. Because it's going in, we also call this afferent. Whenever you see afferent, you know it's going into something. Now, that information is going to be moved into the central nervous system where we are going to go through an integration process. So we're going to take that sensory information and we're going to integrate it into something that makes sense. And then we are going to decide what we're going to do about it, and we're going to send that information out on the motor division. Whenever something is going out, it's called efferent. So let's take an example here. Let's say you see a glass of water, and that information from your eyes goes into the central nervous system, and your central nervous system checks at this information, integrates it with other things, checks your osmolarity of your blood, things like that, and says, yeah, no, I think we should take a drink. So a decision has been made through the integrative process. Now we need to coordinate the muscles that are going to grab the glass of water, bring it up to your mouth, put it into your mouth, and then all of the muscles that are going to help you swallow the water. that is a motor plan that would go out on the motor division. So you've got some type of sensory information coming in, a decision-making process, and integration that occurs in the central nervous system, and then whatever that plan is, it's going to move back out on the motor division. So let's talk about this tissue for a second. It's very, very cellular. When we look at nervous system, we don't see a lot of extracellular space. We see a lot of cells, and there's two different types. We've got neurons, which are your functional cells, the cells that are actually doing the job of running everything else. And then we've got neuroglia, which are the supporting cells. We have a lot of those. In the central nervous system, we're going to talk about astrocytes, microglia, ependymal cells, and oligodendrocytes. In the PNS, we're going to talk about satellite cells and Schwann cells. When we look at this tissue, it's roughly 10% neurons and about 90% neuroglia. So we have a ton of cells in here, but they're mostly neuroglia. The functional cells are neurons, so we're going to start there. We're going to talk about them. So neurons are a cell. They are kind of a weird looking cell, but they're definitely a cell. So we're going to expect to see normal cell stuff going on. But one thing to remember is that neurons can be very long. So when we think about a motor neuron, for example, that is going to innervate your muscles on the bottom of your feet that wiggle your toes, the cell body of that motor neuron is up in the sacral region of your spinal cord. And the axon that comes off of it goes all the way down the back of your leg and under your foot. So there's a process in that coming off of the cell body that is the length of your leg. So you want to keep in mind that these can be really, really long cells. So they're very, very specialized. They have a very specific function, which is to pass information from one cell to another through the electrical excitation. They live for a very long time. You make all of the neurons you're going to make when you are a fetus. In fact, you make more than you need, and then you go through a process called neuronal pruning, where you start killing off some of them. And then that's all you get. You don't regenerate your neurons because they are amitotic, meaning that they do not undergo mitosis. And if we don't have the cell cycle, then we don't divide. These guys are in a sort of permanent state of G0 in terms of the cell cycle. They have a very high metabolic rate, so this means that they need a huge amount of oxygen, they need glucose, they need to be able to make ATP. We do have structures that are different depending on what type of neuron we're looking at. However, there are going to be some similarities among them in that they're all going to have a cell body and they're going to have processes coming off of the cell body. So they're all going to have a cell body, they're all going to have processes coming off of them. Okay. So let's take a look at the cell body here. It's called a soma, a neurosoma, or a perikaryon. So the cell body is where we're going to find all the normal cell stuff. It's the biosynthetic center of the cell. It's where we make all our proteins. We're going to have all our normal organelles in there. Okay, so you're going to have endoplasmic reticulum. You're going to have ribosomes. In fact, all of these little chromophilic substances, or what are more commonly known as Nissl bodies, these are stacks of ribosomes. There's so many ribosomes in there because we have to make so many proteins that they actually show up when we look at the neurons. We're obviously going to have a nucleus. We're going to have the nucleolus where we make all our ribosomes. We're going to have neurofibrils. So this is a network of cytoskeletal elements that are going to help hold this kind of strange structure together. They're very evident when you go down into the axon because we need something to provide support for this incredibly thin projection coming off of the cell body there. When we find cell bodies of neurons in the central nervous system, so when we see this part of a neuron in the central nervous system, it's going to be called nuclei. So this is not to be confused with the actual nucleus of the cell. When we have a collection of cell bodies in the central nervous system, it's called a nuclei. If we have a collection of cell bodies in the peripheral nervous system, it's called a ganglia. So we have different terminology depending on if we are looking at this collection of cell bodies in the central nervous system or the peripheral nervous system. All right, let's take a look at some of this structure here. So here's our cell body, and we can see that we have some short projections coming off of them called the dendrites. We're going to find out that the dendrites are an input zone. The entire nervous system is a one-way flow. So if you go back to the overlapping functions, sensory comes in, integration occurs in the central nervous system, motor input flows out. So it's a one-way direction. The neuron is also one way. Information is going to come in on the dendrites, and it's going to come out on the axon. We're going to find out we can have multiple dendrites, but we can only have one axon, and they're all going to originate from this area called the axon hillock area. And this is the trigger zone for action potentials. So this is going to be an important area a little bit later. Now, running down the axon, you can certainly get branches that come off of it, but you only have one axon coming off of the actual cell body. The axon is going to go as far as it needs to. So like I said, if we're talking about, wiggling your toes, we're talking about something that's at least the length of your leg. And then usually branches, again, toward the end, and then is going to have these terminal components here, and these are your synaptic knobs. You guys have seen this. You've seen this piece right here interact with the skeletal muscle at the neuromuscular junction. So remember that some of this is stuff that you have seen before. Now what we're looking at here is a myelinated neuron. Okay, so it has what's called the myelin sheath on it. We're going to talk about the myelin sheath. We're going to talk about how it's formed, what it looks like. But you can see that you've got the Schwann cells making up the myelin sheath, and between them are areas of bare axon called the nodes of Ranvier. So all of this is going to be important when we get to how action potentials work. But again, it's a one-way flow. Dendrites come in, so you got information coming in, and it's going to go out on the axon, and then eventually it's going to end up at the axon terminal, and we know that we're going to release some type of neurotransmitter, and that's going to then activate our next cell. Okay. All right, so let's talk about the dendrites. We can have various numbers of dendrites here, okay, and they are all inputs. The axon is your one output. So you only have one coming off of the neuron, and then it can branch from there. So the axon comes off of this axon hillock, and the plasma membrane surrounding the axon is called the axolemma. The internal structures are the axoplasm. This very long projection is completely dependent on the cell body. So it's just a piece of the cell that happens to be very, very long. If it is somehow disconnected from the cell body, where we've got all of our protein synthesis and things like that, it's not going to work. So it is completely dependent on the soma here. Now, there is movement in both directions internally within the axon. So a signal, an action potential, only travels down the axon. But if we think about the movement of the neurotransmitters, we'll use that for a second. So neurotransmitters are going to be made here in our cell body, and then they're going to be walked using motor proteins all the way down the axon, and then they're going to be stored in the knobs. So remember the acetylcholine that we talked about with muscles? It was made up in a cell body, and then it was walked in a vesicle all the way down the axon, and it waited there in its synaptic vesicles until the signal came. That's what's called anterograde movement. So we're going to see movement from the cell body to the axon terminals. Retrograde goes in the opposite direction. So let's say we've got an old mitochondria down here in the knob, and we need to get rid of it. It's not doing its job very efficiently anymore. We've got to get rid of it. We're going to grab a hold of it with a motor protein, and we're going to walk our way up the internal structure of the axon and over to the cell body where we can get this thing fused up with a lysosome and get it degraded properly. So there is movement in both directions internally in terms of the interior of the cell, but an impulse, an action potential is always going to travel down. Now, this is where we would find the myelin sheath. So dendrites and cell bodies are not myelinated. Only axons can be myelinated. All right, so let's talk about the different types here. Structurally, we have three types. We have what's called a multipolar, a bipolar, and a unipolar neuron. Multipolar means that there are more than two projections coming off the cell body. Just more than two. That's all it means. Bipolar means you've got a cell body and then you've got two projections. One is a dendrite, one is an axon. Unipolar, here's your cell body, and you have one little projection coming off of it that then connects to this very long axon. So, multipolar, more than two. Bipolar, two. Unipolar, one. Now, the trigger zone, or where we would trigger an action potential, is going to be different in each of these. In sort of the classic version of a neuron, multipolar, it is at the axon hillock area that is very close to the cell body, and then you move into this very long axon. Bipolar, if they have a trigger zone, is going to be right at the base of the axon. And then unipolar is the one that's a little bit different. The trigger zone is right next to the input zone. And this entire thing is actually an axon. Okay, so this one's a little bit different. There's a reason. Okay, so the information is going to flow. It's going to trigger the action potential here. The information does actually go into the cell body, but the signal to release this neurotransmitter is going to get there really quickly because this is all an axon. Okay, so here we can kind of see structurally the differences. So we've got multipolar. Here you're seeing tons of dendrites coming in to a cell body. Here, your cell body, and these are all dendrites coming in. One axon. Okay. Bipolar. Here you can see the dendrite's really short. Here the dendrite's a little bit longer, but we still only have two projections coming off. And then unipolar, you only have this one projection. Here's your receptors, and then this is all axon here. Now, if we talk about where we find them and the overall proportion, multipolar neurons are the most abundant. The two major classes of neurons that we're going to find in this are going to be interneurons, so the neurons that are found fully and completely within the central nervous system. They deal with the integrative processing, so they are the points that connect, the neurons that connect the other things. And then motor neurons. So your motor neurons that are dealing with skeletal muscle, cardiac muscle, glands, all of that, those are going to be multipolar in structure. So interneurons and motor neurons are multipolar. Bipolar are very rare. We're going to find them basically only in the special senses. So eyes and ears are the only places that we're going to find those. So very, very rare. Unipolar are going to be your somatic or your basic sensory neurons. So you've got your receptors out here in the skin, and then remember this is basically all an axon. And then there's going to be your little cell body right before we get into the central nervous system. The reason for this is you want information to travel very quickly into this. Like let's say you're putting your hand down on a hot stove. We want the fastest possible conduction of that information into your brain so that you can make a motor plan to pull your hand away. So the fact that this entire structure is a myelinated axon means it's going to travel very, very quickly. Okay, so now we're going to talk a little about the neuroglia. All right, so here we are in the central nervous system. We got astrocytes. They are everywhere. I like to call them kind of like the janitor cell of the neuroglia. They kind of just do a lot of stuff. So first of all, they're involved in the blood-brain barrier. So you can see that there are these foot processes that come around and they wrap up all the capillaries there. So blood-brain barrier. They interact directly with the neurons. There is indications that they mop up extra ions, that they make sure that the extracellular fluid has the right composition in it. They're just kind of like that janitor that has keys to every single thing and can help you out. Like you don't know what you're doing, go find that guy. He can help you. That's kind of like the astrocytes. They basically just do a ton of random stuff within the central nervous system. Very helpful. Ependymal cells. These are going to line the ventricles in the brain. So you have these cavities in your brain that are open spaces that are filled with cerebrospinal fluid. So here we are in our CSF over here. So these ciliated cells, these ependymal cells, here's all our cilia, they are going to help move the CSF. Then we've got the microglia. These are phagocytic immune cells that help deal with anything that might pass through the blood-brain barrier or any dead and dying tissue in the brain and spinal cord. And then the last one is the oligodendrocyte, which is going to form the myelin sheath in the central nervous system. Okay. In the peripheral, we only have two. We've got the Schwann cells that are going to form your myelin sheath in the peripheral nervous system, and then you have satellite cells that wrap themselves around the cell bodies. Now, the only type of neuron that's going to have a cell body out in the periphery is going to be these unipolar sensory neurons. Their cell bodies are wrapped up in satellite cells that are similar-ish to the idea of an astrocyte in that their job is to simply take care of the cell body, make sure it's okay, and do whatever they need to do to make sure that the cell body is in good working order. So let's talk about the myelin sheath here. The myelin sheath is what differentiates gray matter from white matter. So if you are looking at nervous tissue, oftentimes you can just visually, just with your eyes, no microscope, anything like that, you can see where there's gray matter and where there's white matter. The difference is myelination. Anything that is non-myelinated is going to be considered gray matter. Dendrites, cell bodies, and non-myelinated axons are gray. Myelinated is going to be white matter. The only thing you're going to find in there are myelinated axons. So that is the difference. So now when you begin to look at cross-sections of the spinal cord and you can see the gray interior and the white exterior, you need to start thinking, okay, so that means that all the cell bodies are in the gray. All of the axons are in the white. So make sure that you keep sort of following this information that you're learning, keep dragging it forward into what you're learning next. So let's talk about the peripheral nervous system first. So we're going to have Schwann cells, we're going to have something called a neurolemma, and we're going to have nodes of Ranvier. So first let's take a look at the Schwann cell. So the myelin sheath in the central or the peripheral nervous system is made up of multiple Schwann cells. So this is just one neuron, and you can see 1, 2, 3, 4, 5, 6, 7. There's tons of Schwann cells. So in the peripheral nervous system, you have many Schwann cells forming the myelin sheath for one neuron. Each one of these is a true cell. It has a nucleus. It's got all of the cell components in it. And it is going to bring itself over, and it is going to begin to wrap itself around the axon. So I want you to picture something like about a half-filled tube of toothpaste right now. And I want you to picture taking a pencil at the very end and beginning to wrap your tube of toothpaste around the pencil. Make sure that the lid is on. We don't want toothpaste going everywhere, okay? So you're going to wrap it up. Now you're going to see that the tube is just going to keep forming this layer around the pencil, kind of like what's going on right here. You end up with layer around layer around layer, and this is all plasma membrane. Now what's happening to your toothpaste is that all the toothpaste is getting pushed to the top, okay? The same thing is going to happen with all the organelles and cytoplasm in the Schwann cell. It's all going to keep getting pushed to the outer layer. And that outer layer, right here, you can see tends to be thicker, okay? And that's what's called the neurolemma. This guy right here. This is an important piece of information. The neurolemma is what is going to make nerve regeneration possible in the central nervous system. When we look at the central nervous system, we're not going to see this. Okay, so the neurolemma is a really important piece here. Now, because we have a whole bunch of individual cells here, we are going to have some areas where the axon is showing. Okay, those are going to be your nodes of Ranvier. Those are going to be important as well. Now, in the central nervous system, we have oligodendrocytes. It's going to be set up a little differently. Here, we're going to have one oligodendrocyte that is going to participate in the myelin sheath for multiple neurons. It's the exact opposite of what we see here. Here, we have multiple Schwann cells forming the myelin sheath for one neuron. Here, we have one oligodendrocyte forming the myelin sheath for many neurons. That's a big difference in why we can regenerate out here in the periphery, but we cannot in the central nervous system. Here, your cell body is here. This is just plasma membrane that's basically an extension off of the cell body. There's no organelles out here. There's no protein making machinery. There's nothing. So if you injure something out here, there's nothing in the myelin sheath to help. If you injure something out here, we're going to find out that there is something in there that can help. So here we'll take another look. Here's our single oligodendrocyte, and you can see multiple projections coming off of it that are simply plasma membrane wrapped and wrapped and wrapped and wrapped and wrapped around. Here you have a complete Schwann cell that wraps itself around, and you end up with the neurolemma out here. Okay, so you've got the neurolemma, the basal lamina, and then here's your endoneurium, which is the connective tissue component that surrounds each and every neuron. So if we look at this in cross-section here, here's our axon, and then all of this is the myelin sheath. Okay, it goes around and around and around and around and around. And then here's that layer on the outside called the neurolemma. Okay. Now, here going to point out this is what's called an unmyelinated axon. It is actually still in contact with a Schwann cell. So here it is right here, but it's not wrapped. Okay, so even what we would call a quote unquote unmyelinated axon is going to have an association with the Schwann cells, but it's different than what we see here. This is going to have an insulating property, this wrapped component, whereas this does not. Okay. So let's talk about this concept of regeneration. Your nerve fibers out in the peripheral nervous system are very subject to injury. So, you cut yourself, you have, crush injury, something like that. There is often trauma out in the periphery. Now, remember that the cell body's here. So here we're looking at a muscle. So here's our neuromuscular junction, it's a motor neuron. These cell bodies are going to be in the central nervous system. So what we're talking about is we've got some type of trauma here. Okay, we've cut ourselves, something like that. So what's going to happen is you're going to have macrophages, which are immune cells that are going to come in and they're going to start eating all of this up. Okay. Now, the cell body and the muscle both rely on two-way communication with each other to keep both of them healthy. So as we begin to degenerate all of these cells and the nerve process that was running through them, the cell body is going to start looking a little messed up, and the muscle fibers are going to begin to atrophy. They're going to begin to get a lot smaller here. Now, the neurolemma is going to have a nucleus. It's going to have protein synthesis machinery. It's going to have a whole bunch of stuff, and it can start actually, the Schwann cells can start secreting growth factors that stimulate the end of this axon to grow. Now, this can only happen if there's enough neurolemma left over and that the basal lamina is there. So it's not a guarantee, but it's a possibility. And when you stimulate the end of the axon to grow, it kind of branches out. And you see you've got kind of this structure going on here where there's a whole bunch of branches. If one of those branches actually hits what's called the regeneration tube, okay, so the Schwann cells are going to be able to regenerate because they've got a nucleus, they can go through mitosis, and the interactions with releasing growth factors get the actual neuron to grow, and we get into the growth tube here, the regeneration tube, then we can begin to reestablish contact. You can see that the muscle fiber is continuing to atrophy. The cell body is not looking good. Okay. Once one of these really gets going in the regeneration tube, the others will just basically retract. And eventually we get a reconnection with our muscle. When that happens, we begin to see the two-way conversation going on here. The cell body recovers. We get connections here, and the muscle can start contracting, and so its atrophy begins to be corrected. This is possible in the peripheral nervous system because of the way the myelin sheath is made. You have all of these individual cells, and so they all have a nucleus. They can all undergo mitosis. They can secrete proteins. They can do all of these things. That being said, it's not a guarantee. If one of these guys doesn't hit the growth tube, this doesn't work. If it hits the growth tube, but the growth tube is a little off and you don't reconnect at the neuromuscular junction, that probably isn't going to work. Even if it does work, there's usually some type of deficit there. How significant could be something you don't even notice, but could be really significant. The other thing is this takes a very long time. So we're talking a few millimeters a day of regrowth. So anywhere from about 3 to 5 millimeters a day of regrowth. So if you're talking about, like, a big neuron in your your leg or something like that, you're talking like 2 years of growth before you really begin to see if you're going to get function back. So it's a very slow process. Now, if you injure a neuron in the central nervous system, so the brain and the spinal cord, this can't happen. There is nothing out there to help regenerate the axon if it is damaged, because the myelin sheath is just a piece of cytoplasm that's wrapped around it. The cell body of the oligodendrocyte is a little bit further away from where the injury would occur. So we do not see regeneration in those tissues. Luckily, those tissues are encased in bone, so they're a little less exposed to trauma than everything that's out in the periphery, but it is a far more significant issue when they are injured because we simply cannot regenerate.